Hello everyone, I'm back again, and this time we will be finishing our lecture under the introduction. So last time we have already discussed about the structure of a virus, the different classification of the virus, as well as the mode of transmission of the virus, as well as its replication. Okay, now what we will talk today is about the replication process in a bacteriophage. Okay, so a bacteriophage is a virus that infects bacteria. So why do we have to study also bacteriophage or bacteriophage? Because bacteriophage are good models of discovering the replication cycle of a virus. Okay, in the life cycle of a bacteriophage, this is divided into two types. First is the lytic cycle, and the other one is the lysogenic cycle. So, what happens in the lytic cycle of a lytic phage? Okay, so the same process occurs just like in humans, that it attaches to the host or absorbed to the host, and then it penetrates. The genetic material is injected, and there will be the reproduction of their nucleic acid as well as the synthesis of proteins that are used as building blocks for making their enzymes as well as their structure. There is an assembly, but the difference now is on the process by which they are released from the host. So in lytic phages, the host is lysed when the new phages are released. That's what we call lytic because the cell will lyse when the mature virus particles are released. Whereas in the lysogenic cycle, the host is not affected. Why? Because in the lysogenic cycle, after the entry of the nucleic acid inside the host bacteria, it is spliced or it is inserted into the genetic material of the host. So the viral DNA is integrated into the host DNA. Okay, so we call this bacteria that carries the viral genetic material as the prophage. Okay, so as the cell replicates, the genetic material of bacteriophage is also replicated, such that all new host cells, all new host progenies carries the genetic material or the nucleic acid of the bacteriophage. Okay, that's why it's called lysogeny because the cell can grow normally. They are not affected. They do not lyse, but inside their genetic material are packaged together the viral DNA. And for some time, when these prophages are exposed to triggering mechanism, they can shift from a lytic to a from a lysogenic to a lytic cycle. So there is some form of induction, such as exposure to radiation, exposure to strong chemicals, exposure to variable environmental conditions. So when we subject the prophages in those conditions, they can shift into a lytic cycle. So you can see that one in the left side of the graph. Now, this one will give you an idea how a phage penetrates the bacterial host cell. So, unique with like T phages, bacteriophages, are its structure that is complex. They have an icosahedral head, and they have this bodies or mid bodies that are helical, and they have the tail portion. Okay, so in the attachment is the tail portion of the phages that attaches to the receptor site in the host. So this one is the major spikes equivalent to the spikes of the human viruses. Okay, so they have spikes which are known as tails, and as soon as they are attached, the middle part, which is helical, stretches or compresses. Okay, so mura siyang barina, no? It compresses such that the tip portion of that will secondarily attach to also small receptor sites in the surface of the bacterial cell. Okay, and after that, the genetic material or the nucleic acid that is found in the icosahedral head is slowly released and injected inside the cell. So in bacteriophages, there are no uncoating. Okay, it's only the genetic material that gets inside the host cell into the cytoplasm of the host cell. So the replication of viruses of eukaryotes can be contrasted to bacteriophage replication. Okay, so these are the following similarities and differences. Na? So in the penetration for bacteriophages, they usually exhibit membrane fusion or phagocytosis. And in the replication, it can be in the nucleus or cytoplasm, or parts of the processes may occur in each part of the bacterial cell. And for the release, generally, the progenes of bacteriophages are released via budding. So taking its host cell membrane along, that can be used as envelope as in the case of eukaryotes, but in the case of phages, the structure is very complex. Okay, so for the summary of the manner by which the viral genetic material undergoes transcription and translation as well as replication. So this slide will show you the seven classes based on the Baltimore's classification, which will show the different pathways of the formation of viral messenger RNA, which is depending on what is the composition of the viral genetic material. Okay, so I have already expounded on that, and this will just simplify which one can be used immediately as a messenger RNA and which one can be used immediately. Like, for example, some of such, like, for example, negative, can be used to convert into its genetic material replication or copying. Okay. So if you have question on the replication, we will answer that one when we talk about viral replication in a separate lecture. Now, what are the consequences of viral cell interaction? Okay, so there will be proliferation. What do you mean by proliferation of the virus? Managhan ang mga virus. And as a result of that, when they are released abruptly from the cell will die because the cell will lyse. So this is what you can observe in infections like cold sore, luas, which is caused by herpes mo nang hapdus kay ng luas because the cell will burst. In genital herpes, for example, sakit kaayo ang mga luas sa inyong kiki. No? Chicken pox, when the macules will burst, is very painful because your cells are actually bursting and eventually the cell will die. In the latent infection, this is another consequence of viral cell interaction. In latent infection, the virus do not replicate. Okay? So for herpes, for example, some herpes viruses go into latency. They hide in the trigeminal ganglia for some time, maybe a year, two, or until you become old. Okay, so another is HPV. What is HPV? Human papilloma virus. So you will not know you have it in your younger years, and then when you are already immunocompromised, when you are already old, then it will manifest the infection and it will get out from their hiding place. Okay, so in latent infection, it can be that the viral genetic material is integrated into the host chromosome, host DNA. Okay? So, or it cannot also be. It will just stay there for as long as you have the living cell. Okay? But most of the time, it's just like producing the prophages. In the case of human, we call that one proviruses or provirus. Now, another consequence of the virus in the cell is persistent infection. Okay, in persistent infection, there are only a few or no symptoms. The patient may have just self-limiting symptoms, makaya ra sa pasyente. Or it can be that they are carrying the virus but they do not exhibit the symptoms. Kani siya is persistent infection is a gradual type of infection in which the virus is replicating. There is an ongoing replication, but the replication is slow. It's just like killing you softly. So, what are examples of these viruses? You have hepatitis C and B, HIV. So it will take some time. Maybe you had been drinking for 25 years and then you get infected on the fifth year, no? Because a, s- a co-alcoholic share it with you via an open cuts, no, during the drinking session. However, you notice it when you are already in ages 20 years after, okay, where the symptoms are already exhibited. So, hinay-hinay siya, and sometimes you cannot notice it, but you are already a carrier of the virus. Now, sometimes persistent infection may cause tissue injury or immune response to viral infection. That's why in some people, persistent infection they may resolve without knowing it. So they're carrying it, and for some time they have developed an immune response, and later it will be resolved. Mawala na siya sa inyong system. Okay? Your body is able to eliminate them through the antibodies that are produced that form immune complexes with this virus, no, and it will be sequestered by your spleen, so you will be cleared. And sometimes you notice that when in infections like viruses that can cause transformation, but it is also observable in persistent infection. Another is oncogenesis. So for viruses that keeps on proliferating and the target is the transformation of the cell that they are infected, then it will end up into developing cells, transforming your healthy cells into cancer cells or tumor cells. So example of that are the HPV, mga warts. Those are although warts are benign, there are form of HPV, like for example, the agent of cervical cancer, no, that is malignant. Or maybe EBV. What is EBV? That is Epstein-Barr virus, no? The agent of some form of Burkitt's lymphoma, I think. So some form of lymphoma, and you cannot recover from the infection, but eventually you will also die if your immune system is very weak. And sometimes you are asymptomatic. You do not know that you have it. You do not know they're carrying it. And it just stays there without even developing. Okay? Example of that is hepatitis G. No? No apparent disease, meaning asymptomatic. You are not actually suffering from an infection. You're just carrying it with you. And it is happy to be in your body, but there is no effect, or there is no evident effect on the host cell or on the humans. Okay, so this one will also show you a summary of this, no, in a graphic presentation. So the possible consequences in animal as well as in human cell, no, from the time the virus attaches to the host, appropriate host, it penetrates, it multiplies, what will happen? So those are the consequences. If that is a tumor producing or tumor producing virus, it transforms the cell. So the effect is you will have transformed cells, no? Human cells. So instead of this, may mo silang ing-ani. Okay? It becomes abnormal. They live, but they are abnormal. Okay. Another is the lysis or the lytic infection, which you can find mostly in acute infection. So the cell will eventually die after the release of the virus. Another is persistent infection, in which the virus keeps on shedding. Okay. Slow release of the virus without cell death, but you are carrying the virus and you can be infectious to healthy individuals. And then the last one is the latency. Okay, latent infection. So the virus present but not causing harm to cell, and later it emerges as a lytic form of infection. Okay, so that's why phages had been extensively studied, because that will also tell us how it is in humans when viruses infects humans. Okay, and then for latent and persistent viral infection. Okay, so how do we differentiate latent versus persistent? So when you say latent, the virus remains your system, in the host, but the host is asymptomatic, at it stays there for long periods of time. That's why if you try to look at the graph, it will take months or years, no, when you have the latent infection. Okay, and when you say persistent infection, the disease processes occurs over a long period of time and is generally fatal. So this is what I told you earlier, that you have killing you softly. No? But we also have a term used to describe viral infection. We have the acute infection. This is always manifested by viruses that takes it is lytic cycle in humans. Now, what about viruses and cancer? So we will discuss more on this, but to just give you an overview, no, the genetic material of oncogenic viruses becomes integrated into the host cell DNA, and that's what you call the provirus. And then there is conversion of proto-oncogenes to oncogenes. And these oncogenes are being activated, thus it transforms normal cells into cancer cells. And then the transform cells increase in numbers. They lose contact inhibition. They have tumor specific transplant antigens, and they express their T antigens. Oncogenic viruses is responsible for about 10% of human cancer. So, example of that, we have hepatitis C for liver cancer, uh, HPV for cervical cancer, and some other malignancies and hyperplasia. And then we have the tumor viruses, no? The one that is exemplified in some form of itong mga hubag ng mga tumor in the neck portion, no? Mga herpes virus that are oncogenic. We have the Burkitt's lymphoma, we have the PML, and many others. We will discuss that one when we go to the specific viruses that can cause cancer. So this is what happened in cancer, no? So what will happen is very simple. It integrates its DNA into the host DNA, and then it codes for the production of transform cells when they are expressed. That's why when these cancer cells are already produced, they keep on replicating without any function at all. So that means if that particular cell has specific function in the in that particular individual or body, it loses its function. Okay? So it will be replaced by the cancer cells or tumor cells. So these are some examples. As mentioned earlier, we have hepatitis C from among the RNA. There is a member of the retrovirus, no, which is also causing cancer, the human T cell leukemia virus or HTLV. This is a member of the retrovirus. And then the one that I mentioned also earlier, the human papilloma virus or HPV, which is known to cause one of the cause causes of cervical cancer, and then EBV or Epstein-Barr virus for lymphoma, Burkitt's lymphoma. And then there's another one that arise after the discovery of HIV. We have the herpes virus. This one is the human herpes virus, no, 8, which this is an addition to the member of the herpes viridae family, no, the agent of Kaposi sarcoma. This is a form of virus that transform the cells of the muscles, no, into a tumor cell that keeps on replicating, but it's just under the skin in the muscles. And of course, hepatitis B, although it's not a higher percentage, can also succumb to liver cancer, just like hepatitis C, especially in fulminant hepatitis. Okay, so additionally, I would like to also mention about subviral agents. Okay, mga virus-like entities. So we have the satellites, or now this nucleic acid only containing entity is known as virusoid. So they contain nucleic acid and is dependent into another virus, no, a helper virus, so that they can get inside the cell. So in plants, uh, we have the hepatitis delta. This is the one also in humans, no, because this one is the one that rides on hepatitis B so that it can get inside the cell. Okay, and it can also explicitly increase the virulence of hepatitis B. Okay, so they have the virusoids. And then we have the viroids. The viroids are mostly plant pathogens, viral pathogens. Like an example, it's a small circular single-stranded RNA which is autonomously replicating on its own. Okay, it has no capsule, no capsid, it's just a and RNA. No? So an example of a disease in plant that's implicated in viroid is potato spindle tuber viroid. No? So this one is a virus. We do not find viroids in humans. And then the last one is prion or prions, whatever you call it, no, and pronounce it. So prions are infectious protein. They don't have nucleic acid, so that means they're intact protein. They're highly heat resistant even if you cook. No, that's why do not eat infected raw meat, no. Oh, so dapat imo siya ikot and highly resistant to heat. This affects the nervous system, the nervous tissues. No? So, what are the examples of the disease that prions? Prions, we can see prions in humans, infection in humans. So we have the bovine spongiform encephalitis or SBE, and SBE in cows are known as mad cow disease, scrapie in sheep, Kuro and Creutzfeldt-Jacobs in humans. No? Okay, so prions, these are actually proteins that are transmissible and can be inherited via ingestion, transplant, or even in surgical instruments that are contaminated with this. Okay, so the target is the brain. It can cause or transform the brain into a sponge. Can you imagine that? So there are researchers that will tell us that prions are the cause of the following spongiform encephalopathies in humans. So we have the Creutzfeldt-Jakob disease or CJ, the Gertzmann-Straussler-Schrinker syndrome, and then the most famous familial, fatal familial insomnia. So the one in which you cannot sleep. And of course, in animals, you have the mad cow disease, the scrapie in sheep. Okay, so normally we have normal cellular prime protein on the cell surface, which is involved in cell death. However, in prion disease, it is transformed into a scrapie protein that accumulates in the brain and form plaques and transform the brain into a spongy form like that. So this is the manifestation in sheep, no? And then this is an example of a section of the brain of a person suffering from CJ, Creutzfeldt-Jakob disease, CJD. So the brain will shrink and there is deterioration. So ma-transform imong brain, mahimo kag vegetable. Okay? So it's actually mutation when it becomes sporadic already in the population. So with regards to the viral diagnosis, there are many ways by which viruses can be diagnosed clinically in the laboratory. So the infection can be diagnosed via the manifestation, the symptoms, the signs. Okay, and in the laboratory we have a lot of methods that we can use to diagnose viral infections. We can do a biopsy, cytologic and histologic examination. We can have direct examination under the electron microscope, and then we can also test for the presence of viral antigens either in the blood via the serum or in the tissues, especially for liquid specimens. And then test also via serologically, we can do antibody testing, looking at the titer of IgM or IgG. So when there is subsistent uh, increase in the titer, there may be an ongoing infection. No? So we can also do isolation of the virus. We can do culture. But for the viral culture, this is very prone to contamination of other microorganisms, like for example, bacteria, fungus, yeast. No? So only special laboratories perform virus isolation because it is expensive to maintain cultures like this. So for those laboratories who are developing vaccines now, they're also the ones doing isolation. We can do isolation via culture, we can do isolation using animal. Okay. And then for the new methods, we have the molecular technique. So we have the nucleic acid testing, the mga RNA amplifications, no? So there are methods now that are available for the detection in liquid specimens. There are also methods now that can be available for the detection in solid and semi-solid specimens, like for example, tissues. Okay. Now for bacteriophages, there are also laboratory assays in that. No? Bacteriophage in the laboratory, the target is mostly not the examination per se, but the production of what we call suspensions of pure bacteriophages for the purpose of using it in phage therapy. Okay, so for the culture of phages, it is not the same as culturing bacteria. No, because in the culture of bacteriophage, you're going to use double layer agar, in which the upper layer of the agar, upper half of the agar is very soft. This is made soft because the bacteria, specific bacteria, and the suspected bacteriophage is mixed with the soft agar so that they can move during the process of infection in the agar. So that's the top layer. And then the bottom layer is a solid agar. So the concentration of the top and the bottom differ. So usually you will be using 0.9% agar for the top layer, and then for the bottom layer we're using mga around 1.2 to 1.5%. Okay, so what you can see in a plaque assay is after a 24-hour culture, because bacteriophages infect cells that are on their growing stage. Now you can see the effect in 24 hours, but during the death phase of the bacteria, they do not usually infect. No? So, because in the mechanism of viral replication, they have to ride on to the to the uh, to the machineries of the host cell, so that maapil pud sila, they can be included in the replication cycle. So they have to infect a cell that is actively growing, but not on its death phase already. So what you can see in a double layer agar, after putting a lawn of bacteria, after putting the bacteriophage in a lawn of bacteria, are this. So what are this? These are kaning mga gagmay which show clearings. These are minute clearings. These are what we call plaques or lawn of clearings. Clearings on the lawn of bacteria. So each plaque is known to arise from a single viral cell. Okay, so you can actually cut this one by one, the top layer, and then you elute the you elute the bacteriophage from that, and then replate it 2 or 3 times for the purpose of purification on the same single host. And then after that, you're going to harvest using the a refrigerated centrifuge. No, you can use ultracentrifuge for as long as it's refrigerated centrifuge. The top layer of your suspension after centrifugation, your supernatant, is the one that you're going to harvest because this is the one that contains the bacteriophage that are pure. And then you can manipulate on that. No, you can actually increase the numbers by replating it on the same host. And that's the one that is exploited now in bacteriophage therapy. So, what are the effects of the physical, going back to the viruses? Now, what are the effects of the physical and chemical agents on the virus? So, we know that we learn about antimicrobial control, and we have specific physical and chemical ways by which we can prevent the virus from getting into our system, or we can use them, we can kill them via these processes, or we can halt their transmission via these processes. So in the laboratory we use heat. So heating 56 degrees centigrade for 30 minutes or a few seconds for 100 degrees centigrade, or boiling, inactivates the virus, some viruses. Okay, so like for example, hepatitis A is very susceptible to heat, no? So, for example, that's why when you go to fast food, if you want to use spoon and fork, make sure that it's hot, okay? The insulator or the heater is turned on, not turned off, because if there is someone who is there as and is a carrier of hepatitis A, you will not get infected because the virus is inactivated. So what do you mean inactivated? Okay, they cannot enter a host cell. So probably during the heating process the protein is degraded. Okay, so it cannot become an intact virion. Okay? Now cold stabilizes viruses. Okay, so viruses are stable at low temperature, even at negative 70 degrees centigrade. However, if you keep freezing, thawing, and then putting again in the ref, and then thaw it again, inito na pod, then ibalik na po sa ref, then they can be partially only inactivated, but they cannot be fully inactivated. Okay? And then, drying. Now, depende, no? Drying, the effect on the viruses is variable. Okay? So, depende na sa klase and the structure of the virus. So, some virus survive well, others rapidly inactivated. Okay? So that when you use a dry oven, for the materials that you use in the laboratory, it has to be heated up to 180 degrees centigrade for 60 minutes, meaning 1 hour. Or if not, you can increase by 200 for an hour, or an hour or 30 minutes. Okay. Um, UV radiation definitely inactivates virus. Why? Because this can cause mutation to the virus. Okay, there is a type of mutation that allows the virus to even deactivate their virulent antigens, now the genes that expresses their virulence. Okay, so that's why in the lab, in the virology lab, we always have UV in the safety hoods, no, because that is a way by which we can irradiate viruses. So I think the effect of irradiation is frame shift mutation. Okay, that's the strongest effect of radiation. And then for the chemicals, the effect of chemicals and viruses, we can use organic solvents, like for example, ether, chloroform. We can use that one, especially for specimens, no? Specimens. So for viruses that contains envelopes, they are easily inactivated. But for viruses that are naked or they do not have envelope, they are resistant to ether, chloroform, and other organic solvents. Now, how about oxidizing and reducing agents like formaldehyde, chlorine, iodine, hydrogen peroxide? Most viruses are inactivated. That's why uh, if a patient died of HIV, for example, it is recommended that it has to be embalmed using 40% formaldehyde or formalin. Okay. This is the most effective inactivating agent for viruses, although in humans, formalin at an absolute concentration like 40% is already toxic to human, especially the fumes. No, so that's why in the laboratory just use 10%, but the 10% cannot guarantee that we deactivate the virus, especially if there are many in the tissues. No, chlorine can be. That's why in the lab we immerse, no, in 10% hypochlorite solution, especially the specimens, no, before we dispose it or before you clean and wash the glasswares. And then another one, we have the beta propiolactone. Beta Prolactone mixed with formaldehyde is effective in inactivating, especially if you're preparing for vaccines. So the formaldehyde there helps in the inactivation of the virulence of the virus, but the propiolactone helps in the equilibrium so that the antigens also will still remain antigenic but not virulent anymore. Okay. Now, other chemicals to which the virus are resistant, phenols, mostly viruses are resistant to phenols. That's why we do not usually use Lysol, except for maybe some envelope viruses. No, Lysol. Example of Lysol, that's under phenol, no? That's 3 to 5% phenolics. And then other agents that will deactivate virus are some common disinfectants, no? The best disinfectants are glutaraldehyde, no? Glutaraldehyde, like for example, Cidex, no? They are aldehyde, but they have the same function with formaldehyde. However, kani mga aldehydes mango, they have toxic also effect to humans. So, irritating agent, no? Irritant siya, as well as sensitizer. Okay? So, that's why in the lab, we use hypochlorite, except if the surfaces to which you are going to use it because there are spills there of the specimens that is suspected to have or to contain the virus, trapuhan siya og hypochlorite before it will be covered with alcohol. So, basically, when we use alcohol, the virus will not, it has no effect on the virus, no? It has no effect on the virus, except for some very susceptible ones that are enveloped. Pero most viruses are resistant to alcohol. Okay, so if you notice here in the oxidizing agent, there is no list of alcohol there. Okay, so that ends my discussion in the introduction. Okay, so try to look at your schedule of activities in the introductory part of the subject so that you can be guided as to what will be the next lesson that we have. Now, announcement, this coming Thursday and Friday, I will be giving you a quiz. So please prepare because we will have a quiz about the first lecture. So the first lecture is all about history. And then after that, I will be giving you an assignment that of which you do not immediately pass during that activity after the quiz. So our quiz will probably last for maybe 15 to 25 minutes, and then after that I will separate you into groups so that you can work on your assignment. Okay? So thank you very much for listening and good night.